What a great talk. So many pearls, so clinically applicable, so future looking. Thank you, Dr. Dickerson. We move on to the next talk. Professor Frank Tarazi from Harvard Medical School will be speaking to us about the hot topic of Alzheimer neurotherapies and the trials of immunotherapies. Professor Tarazi is the head of the Clinical Neuropsychopharmacology Lab. He is a clinician and a neuroscientist. He heads the Clinical Pharmacoeconomics program at McLean Hospital, Harvard Medical School. He's authored so many papers and is a highly cited researcher and has experience doing clinical trials in so many neurological and psychiatric disorders. I look forward to Professor Tarazi's talk. Frank, floor is all yours.
Good afternoon, everyone, and thank you for the invite to participate in this meeting virtually. I'm Dr. Frank Tarazi from Harvard Medical School, and today I will be talking to you about the latest finding on immunotherapies for Alzheimer's disease. Without going into too much details, we know that the two pathological hallmarks of Alzheimer's disease in the brain are the accumulation of the amyloid beta plaques and the neurofibrillary tangles. And as we see here from the slide, these are the amyloid beta peptides are usually released as monomers. Then they start aggregating together to form clumps of proteins, what we know as the amyloid plaques. These plaques actually, as they become more and more aggregated and bigger in size, they will start impairing the synaptic dysfunction or causing or resulting in synaptic dysfunction, which eventually will result in neuroinflammation and neuronal death. The other pathway, which we also know about the neurofibrillary tangles, is the tau protein. The tau protein will start aggregating as the p-tau, phosphorylated tau, forming the fibrils and tangles, and also these clumps of toxic proteins will eventually result in the same pathway, which is synaptic dysfunction, followed by neuroinflammation and neuronal death. So the idea of the immunotherapies to develop specific monoclonal antibodies is to try to reduce this aggregation of amyloid beta plaque, or to reduce the aggregation of the tau tangles. And this is accomplished by developing, again, monoclonal antibodies that are specifically raised against these two toxic proteins. So in this regard, we have three different monoclonal antibodies that have attracted attention recently, and one of them, as we will discuss, have shown enough benefit to be FDA approved. And there are two other monoclonal antibodies that are in late stages of development. So we have the first one, aducanumab, then donanemab and gantenerumab. And as we see from these slides, basically, these are the clumps of toxic proteins, whether beta amyloids or tau tangles. And the monoclonal antibodies come and try to bind to these toxic proteins and try to remove them from the brain and expel them out. And by doing so, we will avoid the cascade of events that eventually leads to the neuroinflammation and neuronal death. So I'll start first by discussing the results of aducanumab and the data of this monoclonal antibody. This is an antibody that was developed, or at least right now is under the auspices or management of Biogen, a company in Cambridge, Massachusetts. And looking at what is aducanumab, it's a monoclonal antibody. And as we see, this is the antibody, and has a shallow compact epitope that contributes to its selectivity for A beta aggregates. So the idea of raising monoclonal antibodies is to be very selective and specific to the target of interest. So we will minimize the binding of this antibody to non-specific sites, which may trigger adverse events or side effects. And the antibody will interfere with the aggregation that we have with the fibrils, and reduces the formation of the neurotoxic A beta oligomers. So the trial that Biogen did two trials. that the first one was called the Emerge trial, and it included 1638 patients. They were randomized into 3 arms: the placebo arm that had 548 patients, and then they tested 2 doses of aducanumab. The low dose was, between 3 to 6 milligrams per kilogram, and the higher dose was 6 to 10 milligrams per kilogram. And, as we will see, the titration was needed to reduce the incidence of adverse events. And there, they had the numbers were more or less randomized equally among the 3 arms. From looking at the demographics of these patients, they are, again, the randomization is very much comparable across the 3 arms. Starting with age, they're all around 70 years old. And, looking at the ApoE carriers and non-carriers, also the randomization was very equal in this regard. So we had equal number of carriers and equal number of non-carriers that were, randomized equally across the 3 arms. More importantly, if we look at the baseline disease characteristics and, specifically the scales that we typically use to assess the behavioral symptoms of Alzheimer's, and as we see here, the CDR-SB, which is the clinical dementia rating, sum of boxes, the score was around 2.4 to 2.7 to 2.5 for the 3 arms. A score of 2.4 or 2.5 on the CDR-SB suggests that these patients are in a very early stage of the disease. They have, mild Alzheimer's or even MCI that precedes Alzheimer's. The same for the ADAS-Cog and ADCL, which measures functional activity, as we see. And again, the MMSE, the Many Mental State Examination, they all had a baseline score of 26.4, which suggests very early stage of the disease. This is the idea, that this trial focused on enrolling patients in a very early stage of the disease, either MCI stage, the mild cognitive impairment, or in early dementia or early Alzheimer's. So these patients were treated with these 2 doses on monthly basis for 78 weeks. So trials with Alzheimer's disease tend to be much, much longer, say, than trials for new drugs in depression or schizophrenia, where a period of 6 to 8 weeks will be sufficient to get an idea if the drug is working or not. Here, as we see, after the trial took 78 weeks, and at the end of 78 weeks, we see that patients, especially who received the highest dose of aducanumab and reached the 10 milligram per kilogram, that this high dose, resulted in slowing the decline in cognitive functions by 22%, as measured on the clinical dementia rating sum of boxes, or the CDR-SB. If we look at the other scale, the next scale to look at is the ADAS-Cog, and similar to with what we have seen in the CDR, those patients who were treated with the highest dose actually had a 27 decline in their cognitive functions at the end of 78 weeks. So this is ADAS-Cog 13. It's the Alzheimer's disease assessment scale that focuses on the cognitive subscale of 13 items. And this is widely used in the assessment of cognitive functions and memory and executive functions in Alzheimer's patients. And it's typically one of the standard endpoints that are widely used in clinical trials for Alzheimer's disease. The third one was, the ADCS, which is the Alzheimer's disease cooperative study, the activities of daily living. And this specifically looks at, the daily functional activity of the patients, the degree of independence, the degree of the ability of the patient to engage in functional activity, and, shows that, say, how is this patient able to walk alone, dress alone, eat alone. So this is a very important, usually secondary endpoint to look at the improvement in the functional activity of these patients. And here we saw the biggest effect, that the highest dose of aducanumab resulted or slowed the functional decline by 40%. This is very impressive, in terms of the ability of this treatment after 78 weeks to profoundly improve the functional decline that is typically seen, say, compared to placebo patients. We see an improvement of 40% in their daily functional activity. So these are the three scales that we looked at: the CDR, the ADAS-Cog, and the ADCS are all measuring behavioral endpoints. Reflect different domains of cognition, memory, executive function, daily functional activity. So what about, the biomarker that is supposedly is the target of aducanumab? And in this regard, we want to validate that aducanumab does engage with the target of interest. It does reduce the levels of the beta amyloid, the toxic protein in the brain. And eventually, as a result of this reduction in the protein, we do see this improvement in the different behavioral symptoms that we just saw. So in this regard, you're looking at this, if you look at the left, this is the baseline showing the accumulation of beta amyloid at baseline for those patients who were treated with different doses of aducanumab. Then on the right, where I'm here, is after 1 year of treatment with the antibody. As you can see here, if you compare, the right to the left sections, there is dose-dependent decrease in amyloid plaques. And at the highest dose of 10 milligrams, there is a profound reduction in levels of beta amyloid after 1 year of treatment or infusion of this antibody. So this, the PET scan here does provide evidence that that antibody is engaging with the target, is reducing the levels of this toxic protein in the brain, and this eventually may have translated into the improvement of the behavioral symptoms of the disease. This is clearly shown now that both the low dose and the high dose did reduce the levels of amyloid, it did reduce the levels of amyloid protein, toxic protein, as early as 26 weeks. That we see here significant signals, and it was both doses, the low and the high doses, were effective in this regard. And 78 weeks, we see profound reduction in beta amyloid in using both doses, the low and the high dose. So here we have two things that this trial showed to us. It did show that there are improvements on different behavioral endpoints, and it did show that there is significant reduction in the biomarker that we are going after, which is beta amyloid. Interesting that when we looked at other biomarkers, it was not only beta amyloid that was reduced with this antibody. If we look at the second biomarker that is also involved in the pathophysiology of Alzheimer's, and specifically in the formation of the tangles, the tau, we see here that there is also reduction in tau levels in the CSF of these patients, the phosphorylated tau, and as well as the total tau. Especially the phosphorylated tau has reached significant levels in terms of reduction compared to placebo, and even at lower doses, we do see that the antibody was effective in clearing the levels of tau in the brain. So what we see here, that the antibody provided us with an evidence about engagement with biomarkers. It provided evidence about some improvement in the behavioral symptoms, as we discussed on different behavioral endpoints. So what about the safety of these antibodies or aducanumab? There is something of concern when we do trials with the monoclonal antibodies for Alzheimer's disease. It is called what we call ARIA. And ARIA is the amyloid-related imaging abnormalities, which we look at different potential adverse events that may happen in the brain as a result of the administration of these antibodies. So they can be, the ARIA, the edema, the hemorrhage, the cerebral hemorrhage. So these are all side effects that, may happen with these patients. And that's why, as we will see, that these patients will need to be monitored closely throughout the course of treatment with this antibody. What stands out in the side effects, that at the high dose, the 10 milligrams per kilogram, we saw a relatively high incidence of ARIA-E or edema. 35% of these patients did have ARIA-E. And the second side effect was the ARIA-H, the hemorrhage. 20% of these patients also experienced or had, potentially signals of ARIA-H. If we look at, in general, those were the two most common side adverse events, the ARIA-E in 35% of the patients, followed by ARIA-H with 20%. But then that's something important to emphasize, that the majority of these patients who experienced ARIA-E did not experience symptoms during the ARIA-E episode. So those were either very mild or asymptomatic, and they will only detect it through the frequent and regular MRI scans that these patients had to go through throughout the course of the trial. And the other thing also, that this ARIA-E episode generally resolved within 4 to 16 weeks with no long-term clinical adverse events. So, yes, that this antibody in this trial did cause adverse events, but to a certain degree, they are well managed, especially if detected early through the close monitoring of these patients. So this is the first trial that Biogen did, the eMERGE trial, and from the name, we can say that this trial emerged successfully in meeting the endpoints, in engaging with the biomarkers, and in relatively having a more or less an acceptable adverse or safety profile. They went and did another trial, which is, simultaneously. So they were running two trials, one the eMERGE trial and the ENGAGE trial simultaneously at the same time. And also in this trial, they had over 1,600 patients, 1,647, that they were also randomized across 3 different arms: the placebo, the low-dose aducanumab, and the high-dose, similar to what we have seen with the eMERGE trial. So you can say that the ENGAGE trial is a carbon copy, really, in terms of the design, in terms of the enrollment of patients, in terms of the inclusion criteria, in terms of the doses of the antibody, as well as in terms of the duration of the trial. But here, and as we see here, again, emphasize that even at baseline, all the scores of the behavioral scales, the CDR, the ADAS-Cog, all reflect, as well as the MMSE, all reflect early stage of the disease, either MCI or mild stage of Alzheimer's. And in this regard, even these scores are very comparable, and relative to what we have seen with the eMERGE trial. So in this regard, again, the inclusion criteria for these patients were very similar. Now, unfortunately, the outcome of this trial was completely opposite from what we have seen with the ENGAGE trial. And so we did see that the high dose of aducanumab was able to show some improvement in terms of slowing the decline of symptoms on CDR or an ADAS-Cog, and showing some significant improvement on the daily functional activity. What we saw here is actually there was no improvement on any of the main scales that we use to look at improvement in the cognitive functions or improvement in the memory and executive functions. So here with CDR, there was in fact, 0.03, no difference, no significant difference, no improvement on the 3 main behavioral scales that looked at the different behavioral symptoms of Alzheimer's. And even with the ADCS, which showed profound improvement, there was actually not a single significant improvement compared to the 40% improvement in daily functional activity that we have seen in the eMERGE trial. So, what we see that this trial actually, the outcome was very negative. It did fail on every endpoint that was looked upon or used as to evaluate the efficacy of the drug on the behavioral level. The 3 main scales, as shown here, have failed to achieve any statistical significance, and even the functional activity of these patients was not significantly better than patients who, received, or, the minor improvement of 18% compared to the 40% that we have seen with the same dose and, in the eMERGE trial. So this is about, this is considered a failed trial, the ENGAGE trial. What about the engagement with the biomarker? That was interesting because we saw that actually the 2 doses in this trial did also result in improvement or in reduction of beta amyloid using amyloid PET. And the results were very much similar in this regard in the as what we have seen in the eMERGE trial. And as you see, there is as early as 26 weeks, there is significant reduction in beta amyloid, and that these effects extended even as we see here more significant reduction at week 78. And similar to the EMERGE trial, the effects of aducanumab in the ENGAGE trial was not only limited to the reduction of beta amyloid. As we see here, there was also significant reduction in phosphorylated tau at both the high dose and the low dose in the ENGAGE trial. So the similarities between the EMERGE and the ENGAGE trials really has to do more with the changes in the biomarker. Both trials resulted in significant reduction in the biomarkers of interest, whether with a reduction in beta amyloid or clearance of tau, but they differ in the behavioral outcomes of these trials. So the EMERGE trials did manage to achieve significance on number of behavioral endpoints, as we have seen on CDR, on ADAS-Cog, and on ADCL. Whereas the ENGAGE trial failed to show any improvement on the behavioral symptoms of the disease. So based on these trials, which as we see, were a little bit contradicting to each other, at least when it comes to the behavioral symptoms, Biogen did submit the application for FDA for approving the drug. And in June 7, the decision of the FDA came and they approved, of last year, June 7 last year, the FDA approved aducanumab, and it is traded now under the name of Aduhelm. And it became the first drug actually approved for Alzheimer's disease in 18 years. The last drug approved for Alzheimer's was memantine or Namenda, or in some countries is traded under the name Ebixa. That was in 2003. So now after 18 years, we had the first drug approved for Alzheimer's, but aducanumab can distinguish itself from other drugs as it is now the first disease-modifying therapy for Alzheimer's disease. The approval of the FDA was surprising perhaps for so many, because not just because the outcome, which many people expected that the drug will not be approved, it is because of the regulatory pathway that the FDA adopted to approve this drug. They use what we call the accelerated approval program. And this program, usually the FDA uses only to speed up the approval of drugs that treat serious conditions and that fill an unmet medical need. So really the approval here that was based on the change in the biomarker, and in this case, it was the reduction of beta amyloid plaque in the brain, not on the improvement in the behavioral symptoms. The consistent signal that came out of the two trials, the EMERGE and the ENGAGE, as we have seen, were this consistent reduction in these biomarkers, whether it was the beta amyloid or tau in both trials. But we did not see, more or less consistent signals in improvement of the behavioral symptoms of the disease. So this was questionable. So the FDA used what is typically used in, say, the approval of drugs in oncology. The changes in the biomarker became the primary endpoint that they based their decision on in approving the drug. But then the FDA did not stop that, said, though we have approved the drug at this stage and it can be administered to patients, we have asked Biogen to conduct a phase 4 trial to confirm the anticipated clinical benefits of the drug. And if the trial does not verify that the drug has clinical benefit, the FDA has the right or can remove the drug from the market. So, what we have here is just not a full, complete approval that we typically see with other drugs. It says say a partial approval, the drug is approved, but still Biogen has to conduct an additional clinical trial to validate the therapeutic benefits of the drug at the different behavioral endpoints. And if such a task or an endpoint was not met from the ongoing trial, then the drug can still be removed from the market. And right now, Biogen is conducting this phase 4 trial, which will take still a few years to conduct, because as we have seen, trials in Alzheimer's disease is a long process, will take years to accomplish from enrollment of patients all the way to analysis of data and the release of the results. So this is the, no matter what, June 7th is still, I consider, a turning point in the treatment of Alzheimer's disease, because we finally had the first disease-modifying drug, and we had, the first drug approved in general for Alzheimer's in 18 years. And, this is how the drug is now being given to patients. We said, the FDA, after they revised their initial approval, and said that at this stage, we have to identify patients only with mild cognitive impairment or mild dementia. So this drug is not for every Alzheimer's patients. It's not, I think, useful for patients where the disease has progressed to moderate or severe stages. It's only we have to identify patients in a very early course of the disease in MCI or mild dementia. The drug is given as an IV infusion slowly over a course of 1 hour. And the recommended dose that we should target is 10 milligrams per kilogram. But this drug has to be titrated very slowly. And as we see here, the course of titration, the infusion and this drug is given again once every 4 weeks. So the first, 8 weeks, they will get 1 milligrams, and we slowly titrate out until we reach, the seventh infusion, which is after 7 months. This is when the highest and the optimal dose of the drug will be given after 7 weeks. The idea here with this slow titration is to minimize as much as possible the incidence of ARIA and reduce the risk of ARIA-E and ARIA-H. It's not just that, and how do we know about that? The FDA also requested that these patients need to be monitored throughout the course of treatment by sequential MRIs. So there will be an MRI done prior to initiation of treatment to get a baseline for it. And then there is a second MRI, the seventh infusion, which is exactly the first before starting the first dose, the highest dose, the 10 milligrams, and then a third MRI at the end of 1 year, which is the sixth dose using the 10 milligrams. So, and I think, if the patient is going to extend treatment beyond 1 year, because that's the protocol that was adopted in the label, these patients still need to be monitored very closely throughout the course of treatment with this antibody. So we have the new drug. So what was the reception in the medical community about this new monoclonal antibody? So, the drug, yes, aducanumab was approved, but we have several issues that were raised. Let's not forget that during, the approval of the drug, the FDA did have an advisory committee to evaluate the drug. It was a public committee that in the public domain that basically discussed all the data. And at the end of that, before, this advisory committee voted overwhelmingly to actually reject the drug, not approve it. And, using also people question the use of the FDA of this accelerated approval program to approve a drug for Alzheimer's disease based on a change in a biomarker and not based on an improvement in behavioral symptoms. So because the behavioral data that were presented were not convincing, as we saw that, we had 2 trials, one failed and one succeeded. And, it's not clear at this stage what is the efficacy of the drug in terms of improvement of the different behavioral endpoints. This was followed by that, many academic medical centers in the US declined to offer aducanumab to their patients after carefully reviewing the data. Not just the academic centers, the US Veterans Health Administration, what we call the VA hospital systems, also declined to offer aducanumab to their patients. And when the company tried to get approval for the drug outside the US, the EMA, the European Medicines Agency, rejected the drug completely. And there was, even an advisory committee in Japan recommended to reject the drug as well. And the last straw that were, adding fuel, gas to the fire in terms of acceptance of the treatment was the decision of the CMS in the US, which is the Centers for Medicare and Medicaid. This is the federal agencies that cover most of the health benefits for senior citizens, that they said, that the cost of the drug will only be covered under what they call coverage with evidence development, meaning these patients need to be actually enrolled in a clinical trial before the treatment can be covered. the cost of 28,000. You know, the number may not sound high, but if we factor in, we have over 5 million patients, Alzheimer's patients in the US, and maybe, you know, up to 1 million patients may be eligible for this kind of treatment. This can significantly increase the cost for the treatment using this therapy. So all these things have more or less basically put a dent in the use of clinical use of aducanumab in clinical practice. And what we have seen that the use have been very limited compared to what we have discussed before, you know, about being the first drug approved in 18 years, the first disease-modifying therapy. But, you know, at this stage, people really not questioning whether this antibody is able to engage with the biomarker, is able to reduce, you know, the toxic proteins. It's whether it is effective enough in improving the symptoms, the physical symptoms, the behavioral symptoms of the disease. This actually did not prevent other companies from trying to continue along the same line and perhaps develop, you know, other antibodies that could be even better than aducanumab. And in this regard, Eli Lilly has an interesting antibody right now in development that is called donanemab. And as we see here, the idea that they're hoping with donanemab, they are able to wipe out and clear, you know, Alzheimer's disease from the brain. It's the same, it's, you know, this donanemab, it's an immunoglobulin IgG1, you know, specific from the, for the N-terminal pyroglutamate amyloid beta epitope. And, you know, in phase 1, they found that this antibody donanemab significantly reduced the amyloid plaque, even with a single dose in participants with amyloid positive. You know, so they conducted what you call the Trailblazer study. It's a phase 2 study. And again, they are focusing similar to aducanumab on early stage Alzheimer's patients. And we have seen, this is the baseline characteristics. If we look at the CDR, you know, this is 3.4 relative to 2.6, which tells me that these patients have a little bit, you know, more progression of the disease compared to 2.6. You know, and these patients were, again, no matter what, still they are indicators of early symptomatic population, similar to previous studies, whether with donanemab or whether with other antibodies that were in development. And, you know, they made sure that the ApoE4 carriers were consistent in both, you know, arms. They had, in this regard, they had only 2, not 3. They only tested 1 dose of donanemab at this stage. The number of patients, as I see, we see here, is 272. It is definitely much less than what Biogen had in the both Emerge and Engage trial, where totally there we had over 3,200 patients. But this was still an early phase 2 trial. You can say more or less it's a proof of concept, and a smaller number of patients that were randomized either to placebo or to receive donanemab for, again, a period of 78 weeks. We're going to focus here more interesting, again, on the biomarker, because it's now more and more showing that the bio changes in the biomarker appears to be an important and pivotal step that the, you know, the treatment has to show if, you know, that this is going to follow in the same footsteps of aducanumab and be reviewed under the accelerated approval program. And what I saw with the data that came out of the donanemab, that this antibody was significantly effective in lowering amyloid plaque and plasma tau 217. And as you see here, there's a profound reduction and rapid production of beta amyloid plaques, you know, as early as 6 months. Then the effects were extended all the way to 76 weeks. And, you know, as the same with plasma tau, phosphorylated tau, the reduction starts even earlier at week 12 after 3 months of treatment. And at the end, there is a 23% decrease from baseline, with a total of nearly 30% difference between the placebo and the drug-treated arms in terms of clearance of the plasma phosphorylated tau. Then, you know, Lilly did something interesting, which actually they lowered, you know, as we see here in this study, they stopped the treatment So in this regard, actually, amyloid plaque significantly lowered donanemab treatment at 6 months. And then they stopped the treatment, at 6 months. And they looked what happens for the duration of the trial. And in this regard, there was no build up again of the, amyloid plaques or with tau. So, so this is something, maybe something novel in terms of the treatment, that these patients do not need to be treated on monthly basis throughout the course of treatment. They don't need to be treated for months and years with the treatment. It's enough, if the patient can be treated, especially those patients who have lower accumulation of amyloid plaques at baseline, that it's enough to treat these patients with the drug for only 6 months, as we see here. Then we can stop the treatment, and the effects or the clearance will continue throughout, and after 1 year, even after stopping at 6 months. So that's something interesting. So the course of treatment with donanemab, if it continues to be replicated on a bigger scale trial, can be only for specific patients, for again, that have low accumulation of amyloid plaques at baseline, only for 6 months. Then we can stop the treatment, and we can still follow the behavioral improvement of symptoms through longer than that, because we saw that the effects were sustained that up to 1 year after the termination of treatment at 6 months. And in doing so, we are definitely reducing the risks of ARIA and reducing the need to monitor these patients after the treatment has stopped, and definitely will also result in reducing the costs of treatment. So as we see here, that the effects were very clear, that this antibody is profoundly and rapidly improves the clearance of both the biomarkers, both targets, and effectively engage with these two targets with the much rapid onset of action, and with much profound clearance of beta amyloid and phosphorylated tau. Excuse me. So what happens, and then at the behavioral endpoints, and they used different behavioral endpoints. Eli Lilly has typically developed or devised this scale that we call the IADRS, which is a combination of the ADAS-Cog13 and ADCS. And this was their primary endpoint, and they showed that the drug resulted in 32% slowing in decline of their cognitive function. Looking at other scales, they also range between 23 to 40% decline in the improvement or slowing in the decline of cognitive function. But that's something, though it did achieve 32% significance at week 78, it did not correlate exactly with what we have seen with the profound improvement or clearance of both biomarkers. The clearance can have reached close to 100% in many patients. The improvement of behavioral symptoms was only around 32% on the different behavioral endpoints, especially the primary endpoint using the IADRS. So I think this brings up the issue about the correlations between the change in a biomarker and the improvement in the behavioral symptoms of the disease. It doesn't seem that this correlation is one-to-one. Though we get significant and profound reduction and clearance of these toxic proteins, the improvement in the behavioral symptoms, does not correlate directly. And many times what we see is a lower incidence of or percent of improvement compared to the much higher improvement in the clearance of the biomarker. But this is still very promising in terms of the effectiveness of this antibody, in terms of clearance of the both biomarkers, and in terms of the rapid onset of action. So in terms of the safety and tolerability, again, ARIA-E was an issue. That 27% of the patients did have ARIA-E, which is similar to what we have seen with other agents like aducanumab. Treatment discontinuation was, some very low, depending upon strict protocol criteria, and only 8% had infusion-related reactions. So overall, still the safety is acceptable in this regard, though I still think that these patients, even if they are on Gantenerumab, they will be required to be closely monitored by sequential MRIs. Now, Lilly is conducting a much bigger trial because we said that the Trailblazer trial had only 272 patients, which is a relatively small number of patients. It's okay for a proof of concept, but not okay for registration and getting FDA approval. And so they're having now a much bigger trial. They call it the Trailblazer 2. And they have increased the number of patients from 272. It's going to now enroll up to 1,500 patients. And they are going to use two biomarkers that will be used as a part of the inclusion criteria. They want to make sure that all patients will have amyloid positive as determined by PET scans. And they also have to be stratified for intermediate or high tau burden or intermediate or high levels of tau. And the primary endpoint is going to be the iADRS, similar to what they had used in the Trailblazer 1. This is an ongoing trial. We still will need a couple of years before it is finished, but I think we may get earlier data about the clearance of the ability of this antibody to clear the levels of these two biomarkers before we get the final outcome of the results in terms of the improvement on the different behavioral scales. But it will be interesting to see whether the promising results that we have seen in phase 2 can be replicated in phase 3, especially when using or enrolling a larger number of patients. The third antibody that is right now is attracting attention is called Gantenerumab. It is developed by Roche, and Roche is hoping that this antibody will be able to reconnect the dead wires or the sprouting and connections between different neurons throughout the brain. It's similar to the two previous antibodies that we have seen, that Gantenerumab was also effective in reducing amyloid plaques in patients with prodromal to moderate Alzheimer's disease. And so, for example, if we look at patient number 3, these are scans for 5 different patients. Patients number 3, this is the baseline that look at amyloid plaques, and by the end, after 1 year and after 2 years, there is a profound reduction in beta amyloid after long-term treatment with Gantenerumab. The same applies on all 5 patients. So this drug antibody, similar to the previous 2 ones, also effective in engaging with the target of interest, is effective in clearing beta amyloid in patients with prodromal to moderate Alzheimer's disease. And it did show some, potential improvement in the behavioral symptoms, though the trials were not, that promising. But at this stage, Roche is conducting 2 big phase 3 clinical trials. They are called, the Graduate 1 and Graduate 2. And as we see here that, they have around 1,000 patients in each trial, which is a significant number. And the difference between the 2 other trials that I want to highlight about this antibody, Gantenerumab, that the trial is going to go for 27 months, which is way longer than the 2 trials by Lilly and by Biogen, aducanumab and donanemab. So this will give us an idea about the long-term treatment of using these, monoclonal antibodies. What will be the long-term prognosis? What will be the long-term adverse safety profile, tolerability? Are we going to see new adverse events after, the typical treatment of 78 weeks? So, this is one thing about the, potential outcomes, that to see the long-term, perhaps the benefits or side effects or adverse events after treatment. So the trial will go for actually 27 months. And the second thing, as we see here, there was, a 80% reduction in the amyloid after 3 years in open label extension. The second and unique thing about Gantenerumab is that this drug is going to be given subcutaneous. It is the first antibody that will be given or administered in a subcutaneous formulation. relative to two other drugs, both donanemab and aducanumab will require IV infusion, and this usually tends to happen in a clinical facility. But that's something interesting to see whether the subcutaneous administration of the drug will provide an advantage in terms of more convenience, as the drug can be administered at home by a caregiver. It reduces the burden of IV infusion and enables, in general, to reduce healthcare burden. We are expecting the results, pivotal data, sometime during the next half of this year.
Thank you. I stopped that presentation because it's longer than what we expect, and it's still, there's almost 15 minutes more, and it's recorded, but it will be on the YouTube channel for the group. Thanks for Professor Tarazi. It's a nice presentation. He's updating us about all the autoimmune antibodies, and we will be there during the Q&A to answer all